It is organized into two concentric layers of interconnected ganglia, and ganglia is basically clusters of nerve cell bodies, and those two layers are called the myenteric and the submucosal plexuses. The ENS regulates intestinal physiology independently from that of the brain and constitutes a key relay station along the gut-brain axis, which is bidirectional communication between the gut and the brain. Basically what this means is that if you were to remove the gut from the abdominal cavity, it would be able to function on its own. And because of its autonomy and complex circuitry that is capable of influencing local and systemic physiological processes, the ENS is often referred to as the little brain of the gut, or the second brain. Now, we do know that the ENS, or the gut overall, is key in regulating different immune responses, such as motility, or including motility and secretions. But one thing that we, for the sake of this project, primarily focused on is the regulation in context to varying environmental changes. And one way we do that is by looking at circadian rhythms. So circadian rhythms are incredibly important, as they are an endogenous or internal timekeeping system that allows the body to anticipate environmental changes and synchronize behavior to the day-night cycle. Circadian rhythms are governed by a hierarchical network in which the suprachiasmatic nucleus, or the SCN, which is a structure located in the hypothalamus of the brain, it acts as the master pacemaker to synchronize clocks and peripheral organs and optimizes their function at the right time of day. So the SCN is entrained by light, as you can see in the diagram, and it drives different behaviors such as feeding or sleep. However, the ENS is a part of the peripheral system, and peripheral clocks are entrained by feeding. What we can appreciate from this is that, as you see from the diagram, the SCN is resistant to any changes in feeding. This tells us two things. One, that there is a robust pacemaking ability of the SCN, so that any understandings we derive from this is that overall, as the circadian rhythms of the body, we can affirm that yes, the SCN is a pacemaker. However, it does not tell us how this works with peripheral clocks. And the ENS is a part of the peripheral system, so it's like, what's going on between that? And as the molecular and cellular mechanisms underlying the entrainment and organization of peripheral clocks remain to be understood, we do know that the overall molecular mechanism of circadian clocks is generated by a cell-autonomous, transcriptional, autoregulatory feedback loop. Basically, what this means is that there's a feedback loop driven completely by cell functioning. In the mammalian clock, which is us as mammals, there's three main components that we will focus on today for the presentation, which is BMAL1, CRY, or cryptochrome, and PER, which is period. For this case, we do know that BMAL1 is a master transcription factor, and it drives the transcription of the genes PER and CRY. As this occurs, reverbs, which are key regulators in the transcription, repress BMAL1 by binding to its gene regulatory site known as ROR, and this creates circadian oscillations in clock gene expressions. So if you look at the little diagram and you see that there is like an upward slope and a downward slope in the little circle, any of the upward slopes are caused by BMAL1. It's transcribing PER and it's creating the first part of the cycle, and reverb pushes it down, which allows us to see that kind of like sine cosine wave, and it like flows like that. That's basically how the circadian rhythm is formed. From such, we can understand two things. One, BMAL1 is really important for circadian rhythms under the SCN, which is our primary pacemaker, and it has proven to have a relationship with the structure to create these cycles. However, we also do know that the ENS is able to regulate itself independently from instructions from the brain, as it has its own clock. So this drives us to the primary question that we want to answer with this project, and that is if the deletion of BMAL1 also ablates circadian rhythmicity and enteric neurons. So that means if you were to remove BMAL1, would that remove the circadian rhythm of the gut? Because we know that it is able to independently react. This allows us to see the extent of importance of BMAL1 in the body and the extent of the autonomous regulation that the gut is able to regulate. If this important transcription factor regulates circadian rhythms in the body overall, does it also drive that at the ENS? To test this, we remove BMAL1 and analyze the results. Any questions so far? So in order to address this question, first we need to find a way to monitor the expression of that specifically in enteric neurons, because we're focusing on the gut. Professor Joe Takahashi generously gifted us a HER2 reporter mouse, and this mouse expresses luciferase in a Cree-dependent manner. What this means is that we can monitor bioluminescent expression in specific cells. Oh, let me go back real quick, actually. For this question, we hypothesized that circadian rhythmicity will cease upon the deletion of BML1 because we're asserting that it's really important for circadian rhythms overall. So we're saying even though the ENS is independent, we still think that there won't be a rhythm because of the deletion of BML1. And to test this, we're using Cre-dependent mice. And what that means essentially is that we're looking at the bioluminescence of specific cells. So we're looking at specific expression. And in order to do that, we're activating a AAV injection in CAMK2 mice and activating that in enteric neurons. This just basically means that we're using a specific injection targeting specific neurons. And any signal that we're getting from this is reflective of a rhythmic change of period 2 only in enteric neurons. This is a specific mouse that we were gifted for the sake of this project from now, like we started it for this project, and then continuing here on out. This is a genetic model I have on the screen. You can see like we're cutting out the part that we don't want, or we're stopping the original genetic code, and then we're injecting what we want it to do. And then we can visually see this through our culture procedure, visual culture diagram. And basically, the culture procedure requires us to isolate the outermost layer of the gut, or the myenteric plexus, as it contains most of the cellular information that we want of the ENS. We're going to culture it and then monitor the bioluminescence over a series of days. We used a specific click fetal green luciferase, so as you can see in the bottom portion of the diagram, it like glows green. Anytime we looked at our data, it would show that same green. So for our overall protocol, first, after we get our mice, we inject it, we use the AAV injection, then we will have subject monitoring. This is to ensure that before we actually culture the tissue, the mice are growing the way we want them to. So it's like they're not negatively being impacted by external factors, and they're not being subjected to anything that we don't want it to happen through the injection. Then we have the tissue culture and luminescent monitoring at the same time. As my PI and my mentor, and also the lab that we collabed with, which is Shin Namizaki's lab, as they begin the culture process, I would do the luminescent monitoring. So this would have me take tissue samples, put them in vials of the appropriate medium. First, I would baseline it, and then I would homogenize the samples that I get, and then see what data that I get, what are my bioluminescent readings. And surprisingly enough, every different type of mouse has different scales of baseline. We try to keep it for a range of 100, but sometimes like the samples itself can go up to tens or hundreds of thousands. So it's like, by doing this part of it, we're ensuring we're using the right type of mouse. And then after that, we do data analysis. The data that I'm going to show you guys is not exactly what I see. It's going to be the transfer data, so it's easier for visual analysis, and that would be like this. The following graphs reflect our finding for our initial control model. To ensure that this recording is reflective of the in vivo, or while it's alive, molecular clock, we perform the culture procedure at 2 times, both AM and PM, typically 8 hours apart. The bar at the bottom shows a lights on or lights off environment, and what we can appreciate is that moving past the initial baseline, you can see with like the arrows pointing to the initial baseline, if you move past that because of the difference in time, there is a consistent and robust rhythm that we can see from it. For further analysis, I created the phase plots, which are to the right, to visually reflect the peaks of the samples. To create these graphs, I analyze the rhythm and take the first peaks and convert it to cycle over time, or ZT time. And this is just for an accurate representation of the times. It basically is a mean of standardization. So if you are in a different lab, sometimes they have different time zones that they use. So this is a way that we can just standardize the whole thing. And it basically is saying that 0 is the beginning of the daylight phase, and 12 is the beginning of the night dark phase. And what we can appreciate from this graph is that in both the colon and small intestine samples, the phases generally align, which indicate that our culture procedure, there's no external factors that causes a reset in our circadian phases. And for the strongest results, we typically chose to use the small intestine and colon tissue samples, as it reflected the cleanest data that we could actually physically analyze. But I also collected white adipose tissue, SCN, and then also sometimes fecal matter, but it was cool, for cross-examination. So now we're gonna go on to the actual results that I got. To share, we chose 3 models that reflect our findings. For the first approach, we took a neuron-specific BML1 knockout mouse. Basically, this is crossing the model that we got from Joe Takahashi's lab with a conditional BML flux. We're injecting AAV to delete BML1 and report neurons. So only BML1 deficient neurons are being reported. And this is specific to that of the enteric nervous system. And you can see that in the genetic model, like, I don't know how to explain it, but that's basically what's going on. And surprisingly, the results that we got show that the rhythm remains present despite the deletion of BML1. So if you were to analyze the graphs, you can see that while they're not exactly overlapping each other, there is not A, an 8-hour lapse between the time, and B, there's a consistency within the range of where the peak is at. So even though the values itself aren't the same, it follows the same type of slope, I guess you could say. And I believe the lighter color is the control for all of our graphs that I'll be sharing today. And just to affirm that we can actually see these values, you can also notice the same thing in our phase plot, which is showing that both the control and the sample, the variable data for the small intestine and the colon have a general alignment, and there is not an 8-hour lapse. So this is basically telling us that from our first model, the deletion of BML1 does not remove HER2 rhythmicity in enteric neurons. However, we cannot just use one model and base our entire research off of that. So another model we used was a model under the presence of doxycycline, and this is a drug. Under doxycycline, reverb alpha is overexpressed, which causes the complete repression of BML1. So if you look at the little, I don't know what to call those, but there's like the little half circles, that's the use of doxycycline. And just for a refresher, reverb is what causes reverb is what allows for the presence of an actual cycle. BML is what causes the upward slope, and then reverb causes the downward slope. So if there's an overexpression of the downward slope, there won't be you can't allow any BML to be expressed, so therefore it is a suppression of BML1. And from this, we can tell that surprisingly, there is still a present consistent rhythm. It supports the previous data, and we decided to use different concentrations to make sure that it's not something that we're doing wrong. It's that under different concentrations of doxycycline, it's still showing the same results. So we decided to use 10 micromolars and also 100 nanomolars for both the small intestine and the colon samples. And you can see there is a consistent rhythm with peaks. Moreover, the phase plots also reflect such. Because of the influence of doxycycline on the mouse bodies, we couldn't use too many samples. We couldn't use too many mice for this experiment. And then also, if we try to take multiple samples from the same mouse, the tissue would either rip or just it wasn't usable. So this, unfortunately, only has one sample for each type of mouse that we can use for this one. But the results still reflect the same findings. For our last model, we decided to use a global germline recombined BML1 knockout mouse, which is basically saying that BML1 is conditionally knocked out in all cells of the body. So, like I mentioned before in our first model, let me go back to it. In this model, BML1 is only knocked out in that of the enteric nervous system. For this last one, we wanted to see, is that something that's supposed to happen? So we knock out BML1 in the entire body. Yet still, when we're analyzing our enteric nervous system, we can see a rhythm is present for both the colon and small intestine samples. We suspect that there might be a slight shift in the colon, but we're not 100% sure because, again, we could not get as many samples as we wanted to. There is a greater shift, but it's still not 8 hours, so it's not decisive and based on our guidelines. So from this, we can still come to the conclusion that it is very likely that the rhythmicity of enteric neurons remain intact, irrespective to the ablation or suppression of BML1. And this further certifies that the autonomous ability of the ENS, but it leads us to question what the actual transcription factor of BML1 could be. And to approach this, for further research, we've actually started a few before I left. There's a few possibilities that it could explain why and what we want to do to address it. One explanation is that BML1 may still be intact in some neurons. So to test this, we perform base scopes and qPCR, which requires washing the tissues in appropriate mediums. And this takes a really long time. I hated this part probably of the entire lab process because you're just sitting there for 6 hours. I go to lunch, but sit there and wait for the tissue to kind of won't clean itself, I guess you could say, but that's one way you can do it. Another possibility is that reporter negative BML1 intact neurons may be contributing to first-rhythmicity, which we can address using a neurotoxin TTX or tetrodotoxin. And this is really important because the ability of TTX, the significance of TTX is that it decouples the communication between neurons by blocking sodium channels. So it's chemically altering any connections between the neurons. So even if it's present, which is what we didn't want, if it's still there, by using this neurotoxin, we can make sure that there's no communication still being reflected. A final possibility is that our results were just true, like BML1 is not required for HER2 expression in enteric neurons, and we can test this by using a global BML1 knockout model. This is different from this knockout model because this was conditional. It required the use of the injection. For the other one that we're kind of started doing is from birth, we're going to breed a lack of BML1 in these mice. And this will allow us to see, okay, if we look at the enteric nervous system, because we know we can function without the brain, is our results still reflective of the same thing? And if we find that it is true, BML1 is not necessary, we will look at BML2, which is not really researched right now, so we want to look more into that, as it is a paralog of BML1, because it may tie a role in maintaining first-rhythmicity by using a BML2 knockout model, which is like the one we would do, where it's from birth. And that's really what I had for my research. This is some of the articles that I used. A few of them were from Shin Yamazaki, whose lab we collabed with. He has a lot of significant research on circadian rhythm, so it was really helpful for me to understand like what I was getting myself into with this internship. And I do recommend, if you're interested in this, actually reading some of these. I can give y'all links, but it's very interesting. And lastly, I'd like to acknowledge my lab. To the left, that's Professor Yuki Obata. I was under his lab in the immunology department, and then to the right is my mentor, Isabel. They were such a great support team, and I learned so much under them, and I would genuinely love to go back if they let me. And this was my overall poster. I have it here as well. I tried to break it down as much as I could, but yes. And that's really all I had for my research part of my presentation.